The Great Pyramid of Giza has been standing for 4,500 years. It contains enough stone to build a wall around the entire country of France. Its base is aligned to true north with a precision that modern engineers still cannot fully account for. For nearly 5,000 years, the question of who built it has been answered with educated guesses, political arguments, and assumptions dressed up as scholarship. Nobody, until recently, thought to ask what the biology said. When scientists finally extracted DNA from the ancient Egyptians themselves and ran it against every known population on Earth, what came back was not a confirmation of anything anyone had argued. It was something considerably stranger than that. The people who built the greatest monument in human history do not match any living population on Earth. What the DNA revealed does not just change the story of Egypt, it changes the story of us. Stand at the base of the Great Pyramid and the numbers stop feeling like numbers. The structure rising above you contains approximately 2.3 million individual stone blocks. The lightest of them weighs around 2.5 tons. The heaviest, deep inside the king's chamber, weigh up to 80 tons each. If you took every stone in the pyramid and cut it into pieces roughly the size of a standard house brick, you would have enough material to build a path around the entire circumference of the Earth 2.5 times. That is not mythology. That is volume. The base covers 13 acres. Each of its 4 sides measures almost exactly 230 meters in length, with a margin of error between the longest and shortest side of less than 20 centimeters, across a total perimeter of nearly a kilometer. The corners are almost perfect right angles. The entire structure is oriented to true north with an accuracy of 0.05 degrees. For context, the Paris meridian, which took French engineers years of careful calculation to establish in the 18th century, is less accurate than the alignment of a building constructed 4,500 years earlier by a civilization that had no telescopes, no GPS, and no iron tools. The granite lining the king's chamber was quarried at Aswan, 800 kilometers to the south. The limestone casing stones that once covered the entire exterior, now mostly stripped away, were cut to fit with joints so tight that a piece of paper cannot be pushed between them. The standard explanation for all of this is that it was accomplished by large teams of organized workers using copper chisels, wooden sledges, ramps, and rope. That explanation is not wrong necessarily, but it is incomplete in ways that matter, because it answers the question of how the pyramid was built with a very general sketch and then moves on, as though the sketch were sufficient. It is not sufficient, and the reason it has never been sufficient is that the question sitting directly beneath it has never been properly answered. Not with evidence, not with data, only with assumption. The question is this: who were the people who actually did this? Not in the sense of which Pharaoh commissioned it. That question has a conventional answer, however disputed. But in the deeper sense, where did the builders come from? What population did they belong to? What was their genetic heritage, their biological ancestry, the chain of human movement and mixture that produced the specific people who stood on that plateau and put those stones in place? For the entire history of Egyptology, that question has been answered by looking, by examining mummies, by studying art, by reading the physical features of preserved remains, and arguing about what those features implied. The arguments were loud, they were frequently political, and they were almost entirely based on what people could see and what they wanted to see, which are rarely the same thing. What nobody did, for reasons that made scientific sense at the time and seem almost unbelievable in retrospect, was sequence the DNA. The builders left the pyramid. They did not leave their genetics in any database. Until the technology finally caught up with the question. The image of the pyramid builders that has lodged itself in popular imagination is a mass of anonymous bodies, faceless labor dragging stone across desert under a burning sun. That image is wrong. not partially wrong, fundamentally wrong. And the evidence that contradicts it has been sitting in the archaeological record for over a decade, largely ignored by everyone except the specialists who found it. In 2013, a team of French archaeologists excavating a cave system at Wadi al-Jarf on the western shore of the Red Sea pulled something out of the ground that nobody had expected to find: a collection of papyrus documents, not fragments, not partial sheets. Substantial, readable records dating to the reign of K.H. Hufu, the pharaoh credited with commissioning the Great Pyramid. Among them was the oldest papyrus ever recovered anywhere on Earth, and it was a diary. The man who kept it was named Miriar. He held an administrative title that translates roughly as inspector, and he was responsible for supervising a team of workers whose job was transporting limestone blocks from the quarries at Tura across to the Giza Plateau. Miriar recorded everything: departure times, arrival times, how many blocks were moved in a given period, what his team ate, when they rested. The logistical details of a massive, organized, carefully managed operation written down by someone who was living inside it, and found it ordinary enough to document like routine paperwork. This is not the record of slave labor. It is the record of a workforce. A structured, administered, accountable workforce operating within a system sophisticated enough to keep written logs of its daily output. The physical evidence at Giza confirms it. The workers' village excavated south of the pyramids revealed a settlement built to house and sustain a substantial permanent population. There were bakeries producing bread at industrial scale. There were breweries. There was a medical facility, and the skeletal remains recovered from the site show evidence of bone surgery, of fractures that had been set and healed, of a population receiving organized medical care. The workers who died at Giza were buried in tombs, not mass graves, individual tombs with grave goods constructed with a care that indicates these people were valued, recognized, and remembered. They were not slaves dragged from somewhere and worked to death in the desert. They were Egyptians, or at least people living and working within the Egyptian state, fed and housed and buried by the civilization they were building for. Which makes the question of who they actually were considerably more important than the anonymous labor narrative ever allowed. These were specific human beings. They had faces, they had families, they had a genetic heritage that connected them to somewhere, to some chain of ancestors stretching back through thousands of years of human movement. Merer wrote his name in a document that survived 4,500 years in a cave by the Red Sea. His DNA did not survive, but the DNA of people like him, people from the same population, the same genetic community, the same ancestral stock, was preserved in mummies. And when scientists finally extracted it and read what it said, the answer was nothing like what anyone had assumed. The science that finally answered the question did not arrive quickly. It arrived after decades of failure, false starts, and a fundamental technical problem that had nothing to do with lack of interest and everything to do with chemistry. Ancient DNA degrades. That is not a complication or an inconvenience. It is a basic biological reality that shapes everything about what the ancient genomics field can and cannot do. Genetic material breaks down over time, and the rate at which it breaks down is determined largely by the environment surrounding it. Cold slows degradation, heat accelerates it, dryness helps preservation, humidity destroys it, ultraviolet radiation damages it, bacterial intrusion corrupts it. The environments where ancient DNA survives best are permafrost in Siberia, limestone caves in northern Europe, high-altitude burials in the Andes, stable, cold, dry, and largely undisturbed. Egypt looked like the opposite of all of that. Daytime temperatures regularly push past extreme thresholds. The Nile Valley carries humidity that the surrounding desert does not. The tombs themselves, while sealed at burial, had not remained sealed. They had been opened, looted in antiquity, excavated by archaeologists in the 19th and 20th centuries, handled, moved, unwrapped in some cases, and subjected to a century of study conducted by people who were not thinking about genomic recovery because genomic recovery did not yet exist as a concept. Nobody excavating a mummy in 1905 was wearing cleanroom protocols and worrying about contaminating ancient DNA. They were doing the science available to them. The science available to them left a century of modern human DNA layered on top of whatever ancient signal remained. The contamination problem was severe enough that for a long time it seemed effectively insurmountable. Every researcher who had ever touched a mummy since its excavation had potentially deposited their own genetic material onto the sample. Separating that modern noise from a genuine ancient signal required a level of precision that earlier methodologies simply could not deliver. So the field moved elsewhere. Europe accumulated hundreds of ancient genomes. The steppes of Central Asia produced data that rewrote the understanding of Indo-European migration. The ancient Near East began building a substantial genetic record. The science was moving fast, producing results that overturned established theories in real time. Egypt remained a blank. Not because nobody wanted the data. The demand for genetic answers about ancient Egyptian origins was enormous. But wanting answers and being able to recover them reliably are entirely different things. What changed was methodology. Researchers at the Max Planck Institute for Evolutionary Anthropology developed screening processes capable of identifying the specific chemical damage signatures that accumulate in genuinely ancient DNA over thousands of years. Modern contaminating DNA does not carry those signatures. Ancient DNA does. That distinction, applied with sufficient rigor, made it possible for the first time to separate real ancient signal from modern interference with a reliability that earlier approaches could not match. They also identified which tissues preserved genetic material most effectively inside mummified remains. Dense bone, teeth. The hardest structures in the body, least accessible to bacterial intrusion and environmental damage. Armed with those methods, they turned to Egypt, and this time, the DNA was there. The blank on the map was about to be filled. What it contained had been waiting, undisturbed inside those dense tissues, for 3,000 years. The study published in Nature Communications in May 2017 was not a small or tentative undertaking. The Max Planck team, led by paleogeneticist Johannes Krauss, worked with remains from Abusir el Melek, a site south of Cairo with a continuous archaeological record spanning nearly 2,000 years of Egyptian history. They extracted usable genetic material from 90 mummies. Mitochondrial DNA, which passes from mother to child and traces broad maternal lineages, came back from all 90. For 3 of the mummies, conditions were good enough to recover nuclear DNA as well, the kind that is inherited from both parents and provides the detailed population-level analysis that mitochondrial data alone cannot deliver. Every fragment was tested for the chemical damage, signatures that only accumulate in genuinely ancient material over millennia. Modern contaminating DNA does not carry those signatures. The samples that passed screening were the samples that could be trusted. What they showed was immediate and striking. The ancient Egyptians at Abusir el Melek shared almost no DNA with modern sub-Saharan African populations. The genetic distance between the mummies and populations from below the Sahara was large, consistent, and ran without interruption across the entire dataset spanning 1,400 years of remains from the same location. This was not a marginal result sitting at the edge of statistical significance. It was a clear, stable pattern. Across 90 individuals, across more than a millennium of continuous occupation at a single site, the signal held. Instead, the mummies were most closely related to ancient farmers from the Levant, the region covering present-day Israel, Jordan, Lebanon, and surrounding areas. They were also closely related to ancient Anatolian populations and to Neolithic Europeans. These were the genetic communities that the ancient Egyptians resembled most strongly, not populations from the south, populations from the northeast. Then comes the finding that deserves to sit for a moment before anything else is said about it. The ancient mummies were more closely related to ancient Europeans and Anatolians than to the people living in Egypt today. modern Egyptians carry approximately 8% more sub-Saharan African ancestry than the ancient mummies do. The researchers identified this as a later influx, most likely occurring somewhere around 700 years ago, well after the period the mummies represented. Something shifted in the genetic composition of Egypt's population during the medieval period, and that shift is visible and measurable in the data. The ancient population and the modern population are not the same population. The Egypt that raised the pyramids, that produced Ramesses and Akhenaten and Tutankhamun, was genetically distinct from the Egypt that exists along the Nile today. That is not an ideological claim. It carries no political implication about the relative worth of any population. It is simply what the data shows, clearly and consistently, across a rigorously screened data set published in one of the most respected scientific journals in the world. The people who built the Great Pyramid are gone, not just as individuals, which was always known, as a population. The genetic community they belong to has no clean match anywhere in the present. Their closest living relatives are distant, transformed by thousands of years of subsequent movement and mixture. They built the most enduring structure in human history and left almost no trace of themselves in the people who followed them. The Levantine connection did not appear from nowhere. Understanding why the ancient Egyptians were genetically closest to ancient farmers from the Levant requires going back considerably further than the mummies themselves, further than the pyramids, further than the dynasties, back to a period before Egypt was Egypt, before the pharaohs, before the first unified kingdom emerged along the Nile around 3100 BC, back to the populations that were already moving through and settling across the ancient Near East when all of that began. The Chalcolithic period, sometimes called the Copper Age, sits between the Stone Age and the Bronze Age. In the Levant, this period produced something that archaeology had documented but genetics had never fully explained until recently: a specific population, coherent and culturally distinct, occupying the region in a way that left a substantial archaeological footprint, but whose precise origins and movements remained unclear. In 2017, the same year as the Egyptian mummy study, an international research team published the genomes of individuals recovered from a large burial complex near Peki'in in northern Israel. 22 individuals from a necropolis containing the remains of roughly 600 people. What they found was unlike anything documented in earlier or later populations from the same region. These people drew approximately 57% of their ancestry from local Levantine Neolithic groups. 26% came from Anatolian Neolithic populations. 17% traced back to Chalcolithic groups from Iran. The combination was new. It had not existed in the region before this period, and it carried something that earlier Levantine populations had not shown: genetic markers for light pigmentation, fair skin, blue eyes, turning up in skeletal remains from a region not typically associated with those traits in any popular account of ancient Near Eastern history. The community at Peki'in carried recessive genetic characteristics that surprised the researchers precisely because the geography made them unexpected. This population did not persist unchanged. The specific genetic combination documented at Peki'in disappears from the record, but the ancestry it represented did not vanish. It dispersed. It moved. It contributed to the genetic makeup of populations across a wide region during a period when human movement through the ancient Near East was sustained and substantial. The connection to ancient Egypt becomes coherent when you map that dispersal against what the mummy data showed. The ancient Egyptians were not simply related to whoever happened to be living in the Levant at one fixed moment in time. They were related to the deeper ancestral substrate of the entire region, the accumulated genetic heritage of populations that had been moving through and settling across the ancient Near East for thousands of years before the first stone was laid at Giza. Levantine Neolithic ancestry, Anatolian Neolithic ancestry, Iranian Chalcolithic ancestry filtering westward and southward through generations of population movement and mixture until it produced the people who stood on that plateau. This does not reduce to a single origin point. Population histories never do, but it establishes something clearly and without ambiguity. The people who built ancient Egypt carried ancestry that connected them to the ancient Near East, to Anatolia, to the Levant, and to the deeper genetic heritage of populations whose movement shaped the ancient world. The geography of Egyptian origins pointed northeast, not south, not west, northeast, and the DNA confirmed it completely. Egypt is not an isolated case. That is the part that changes everything. The instinct when confronted with findings like the ancient Egyptian genome data is to treat them as a specific story about a specific place, to file it under Egypt, update the relevant chapter, and move on with a slightly revised version of the history everyone already thought they knew. That instinct is wrong, because the pattern the Egyptian data belongs to is not regional, it is global, and it is appearing on every continent where ancient DNA can reach far enough back into the record. In 2003, a rock shelter in southwestern Libya yielded the remains of 2 women wrapped in woven mats, dead for 7,000 years, preserved by geological accident in a natural desiccation chamber that the surrounding desert should have made impossible. It took 20 years and 3 separate research teams to extract anything usable from the material. When a full genomic analysis finally published in 2025, the results followed the same logic as the Egyptian data and then went considerably further. These 2 women carried no sub-Saharan African ancestry whatsoever. None. In a region the standard historical model had placed at the center of a genetic bridge between the populations of North Africa and those below the Sahara, these women had no detectable connection to any population south of the desert. Instead, they belonged to a lineage that had never been documented anywhere, not in any living person on Earth, not in any ancient genome previously recovered from any location. A population isolated in North Africa for 50,000 years, culturally engaged with the world around them and genetically sealed off from it simultaneously. The Green Sahara had been real. The lakes and rivers and grasslands had been real. Cultural exchange had demonstrably happened across that landscape. The populations on either side had not mixed. Culture traveled. Genes did not. The Americas told a version of the same story at a different scale. In 2016, geneticist Bastian Lamas sequenced mitochondrial genomes from 92 pre-Columbian mummies and skeletons collected across South America and Mexico, remains ranging from 500 to 8,600 years old. His team identified 84 distinct maternal lineages representing the full genetic diversity of the ancient Americas before European contact. Not one of those lineages exists in any living person today. 84 separate lines of descent accumulated over tens of thousands of years. Gone. Then, in 2025, came the first ancient genomes ever recovered from Colombia. 21 individuals from the Bogotá Altiplano. 7 of the oldest belong to a population documented nowhere else, matching no known ancient group in the Americas and no modern population anywhere on Earth. By 2,000 years ago, they had vanished entirely, replaced by a genetically distinct group without apparent transition. 3 continents. Different timescales, different causes, the same result. Populations alive for millennia, coherent and culturally distinct, disappearing without leaving a clean genetic trace in the people who came after them. The pyramid builders are part of this pattern, a population whose genetic identity has no match in the present, whose closest living relatives are distant and transformed beyond clear recognition. They shaped one of the most extraordinary chapters in human history. They built the most enduring structure our species has ever produced, and they left almost no trace of themselves in the world that followed them. The survivors wrote the history. The lost populations did not get to. The Great Pyramid is still standing. The people who built it are not, in any sense that genetics can currently locate. Their culture survived in stone. Their DNA did not survive in the populations that followed them. That is not a diminishment of what they accomplished. It is an expansion of it. Because what the science is now revealing is that the builders of the most extraordinary structure in human history were more distant, more mysterious, and more fascinating than 5,000 years of assumption ever allowed. The genetic record for the earliest phases of Egyptian civilization remains almost entirely unsampled. The pre-dynastic period is still dark. Every unanswered question is now a direction. The pyramid has been waiting 4,500 years. The questions the DNA is forcing are not going to resolve quickly, but they are finally the right questions, and we are only beginning to learn how to ask them.